Good afternoon, everyone. My name is Dr. Jessica Ma, and I'm an assistant professor at the Albert Einstein College of Medicine here in New York. Good afternoon. Welcome to my webinar. I'd like to thank the organizers of BioConference Live for the opportunity to talk about my research today. Although this is a webinar, I just wanted to mention that this is an interactive webinar. So I'd really like you to submit your questions both during and after the presentation. I'd really welcome feedback and ideas and questions that you may have. So I'm going to get started. The title of my presentation is Investigating Tissue-Specific Networks of Cancer-Causing Mutations. And basically the starting point for my talk is to step back and consider how complicated we are as human beings. So we know that the human body consists of over 200 highly specialized cell types. And when we look at these cell types, they're incredibly diverse. They differ in terms of morphology, in terms of plasticity, turnover rates, et cetera, et cetera. And what's really remarkable when we consider the amount of diversity present is that most cell types share the same genome, so that the diversity we're actually seeing is a result of epigenetic modifications and transcription factor networks. So if we want to understand what are the genes that are driving the differences that we see between cell types, we really need to start by looking at its transcriptional profile. Now, when we think about cancer, we often talk about cancer in terms of its discrete states, like prostate cancer, breast cancer, lung cancer. But when we really think about it, cancer is a systems level disease. So if we think about us as humans as the system, cancer is a disease that targets different parts of the system. And it's made up of a broad spectrum of diseases that all share in common this aspect of unregulated cell growth. So when you think about cancer as a perturbation of our different organs or tissue types or even cell types, there are over 200 different types of perturbations or different cancer types that affect the human body. Now, when you think about it from this perspective, a question that comes up automatically is, why are some tissues more robust than others at combating these perturbations to the system? So when we think about talking about cancer in terms of breast cancer, prostate cancer, tumors arise in a highly tissue-specific manner. So when we think about germline mutations, a germline mutation is defined as any change in a gene in a reproductive cell, so for instance like an egg or a sperm, that's incorporated in the DNA of every cell type in the offspring. We know that there are about 80 genes with germline mutations that predispose cells to cancer. Breast cancer is a good example of that. Out of all the cancer genes to date, though, we know that about 20% have germline mutations, and about 10% show both somatic and germline mutations. Now, given that germline mutations give rise to a hereditary form of cancer that's present in every single cell type of the body, this is an excellent starting point for understanding tissue specificity of cancer. So just to give you an example, consider BRCA1. So BRCA1 we know functions as a tumor suppressor. We know that mutations in BRCA1 are linked to hereditary breast and also ovarian cancers. As a gene product, BRCA1 is involved in repairing DNA double-stranded breaks, and it's involved in mismatch repair. So mutations in BRCA1 account for an overwhelming number of hereditary breast ovarian cancers. And the interesting thing is that women who have BRCA1 mutations that they've inherited have that mutation in every single cell type of their body, and yet they develop cancer in the breast and or ovaries. So it's an interesting question as to what is occurring in the other organs and the other tissues that are buffering this BRCA1 mutation. another piece of the puzzle is that BRCA1 is highly expressed. It's known to be highly expressed in many different cell types, not just the breasts and the ovaries. And that expression occurs both at the transcript and the protein level. So it's not a simple matter of BRCA1 being deactivated or repressed in terms of expression in these cell types that leads to the different response we see of developing cancer and not developing cancer. So what we wanted to do was answer this specific question: how is BRCA1 normally regulated in different tissues, as a way of understanding what happens in the presence of cancer, why BRCA1, people with BRCA1 mutations only get cancer in breast or ovaries. So what we've done is we've tried to contrast the tissue-specific regulatory networks in different kinds of healthy tissues that involve BRCA1 to really get at the tissue-specific network differences that might allow a cell to buffer a BRCA1 germline mutation. And so really, as I stated, we're trying to understand how some tissues are able to tolerate these germline mutations better than others. And our goal is to reconstruct, based on data resources that I'll explain soon, and analyze these tissue-specific gene networks to understand what the local network components are contributing towards the development of susceptibility or resistance to cancer that we observe in these different tissues. So basically, just really quickly, the kind of workflow that we've stepped through in our analysis is, first of all, to identify the set of cancer-causing germline mutated genes that we know about. We've then gone ahead and found out which of those genes are associated with tissue-specific tumors and also tissue-specific expression profiles. We've then gone ahead and built protein-protein interaction and transcription factor networks around those genes which have been shown to cause tissue-specific tumors and also be expressed across a wide range of different tissues. And then we've gone on and looked at what functional insights can be gained by contrasting these gene sets and these gene networks. So first of all, we used the Cancer Gene Census as a resource for identifying the set of cancer genes with germline mutations. The Cancer Gene Census is a resource available from Sanger. It's a catalog of genes that have been identified as having a causal role in cancer. And interestingly, more than 1% of all human genes have been implicated in cancer via mutation. So this census is regularly updated. It's a really good resource. It contains a lot of other aspects related to cancer, not just whether they're involved in germline mutations. In terms of looking at penetrance, we know, of course, that not all tissues carrying the same germline mutation will go on to develop cancer. Some germline mutations are associated with cancers that are highly tissue-specific, like BRCA1, but there are also others where the cancers affect tissues more ubiquitously. So it's important in looking at this analysis to define a property that we refer to as tissue-specific penetrance, and this reflects the ubiquity of the tumors in terms of the tissues that they affect. And we've actually used a quantitative definition here where we define tissue-specific penetrance as the property observing a larger number of tumors associated with germline mutations that are occurring in one or two tissue groups. And I'll expand on that in the next few slides. So in terms of identifying and assessing tissue-specific penetrance, the resource we've turned to here is to use COSMIC. COSMIC stands for the Catalogue of Somatic Mutations in Cancer. And even though it's got somatic in the title, it actually contains germline mutations as well. So essentially, COSMIC extends the Cancer Gene Census. It contains a lot of information from the literature on cancer samples, including their penetrance and observation in different tissue categories. So COSMIC has declared 40 standardized tissue categories that cover the entire human body. And what we're interested in looking at is essentially coverage of the number of mutations that have been reported for a specific gene and the exclusivity or the tissue specificity that's then observed. So to give you an idea of what I mean, this is the tissue-specific profile for TP53. And it's kind of hard to read, but going across the x-axis are the 40 different tissue-specific categories that Cosmic is using. And along the y-axis is the tissue percentage. So that reflects the percentage between 0 and 100% of mutations that have been reported in P53 that are exclusive to that specific tissue group. So you can see that the tissue percentage bounces around quite a bit across the different tissue categories. And that really reflects the ubiquity of P53 mutations that we know about already. The tissue-specific mean that's showed in red, it's kind of faint, but you can see the points that are across the graph. This reflects the average of all tissue percentages for all genes that are in the Cosmic database. And the reason why this is important is because different tissue categories do have different rates of coverage or different numbers or sizes in the Cosmic database. And I think this speaks to the fact that some tissues are easier to collect than others. So in declaring tissue-specific penetrance, we've used the tissue mean or the global tissue mean that's based off all of the genes in the database as a way of declaring whether a gene is tissue-specific or not. To give you a further idea of what I mean, we can contrast the profile of TP53 with BRCA1. So here you can see that the three tissue groups where the tissue percentage is very high for BRCA1 are actually breast, lung, and ovary, moving from left to right along the x-axis. And what we've done here is we've used our cutoff such to declare a gene that's tissue specific if its tissue percentage profile is higher than the global mean. So you can see here that for breast and ovary, the tissue percentage is higher than the tissue mean, whereas for lung, it's not. And again, I think this speaks to the fact that there are a lot of lung tissues in the Cosmic database. So this is a way to prevent being biased towards that sort of selection that's available. So our question now is, as I mentioned, to find out what network properties that could be predisposing breast and ovary towards developing cancer that are not present in the other tissues that we're looking at. So when we look at all the genes that we have germline mutations for that predisposed towards cancer, there are about 76, I believe, at last count. And we use the criteria such as when we look to BRCA1 to say how many tissues have a tissue percentage that's higher than the global tissue mean. If that number is 1 or 2, then we consider that gene to be tissue specific in its penetrance for cancer. And so when we look at the genes, the 76 genes that have germline mutations predisposing for cancer, we see that most germline mutations are associated with tissue-specific tumors. And here I've got a graph that just shows you which genes fall into the number of tissues genes mutated in. So we see all the way at the end of the spectrum here, we see TP53 that's mutated in 8 tissues. And on the left, we see BRCA1, BRCA2, etc., that are in the lower end of tissues that have mutations in genes that are mutated in. So really what we've got now, we've got two properties of tissue-specific, two tissue-specific properties, sorry. We've got tissue-specific penetrance, like BRCA1 is specific for breast and ovaries. And we also have this concept of ubiquitous versus tissue-specific gene expression. And so our goal is to put these two properties together and consider which genes that are tissue-specific in their penetrance, but highly expressed across different tissues like breast, ovary, and brain, to understand how it is these tissues are buffering the tissue-specific effect. So I should just step back a little bit and explain how we look at tissue-specific expression. Now, the resource that we're using is a resource that came to us via our collaborators in the Fantom5 consortium. Now, Fantom5 is an international project that's being led by the Riken Omics Science Center in Yokohama in Japan. And the goal of the Fantom5 consortium is to construct a complete promoter map that defines all the different tissues in the human body. So as part of this project, Fantom5 have collected over 1,000 human primary tissues and cell lines, and they've analyzed these by mapping the promoters for these different tissue types. And they've done this mapping using a technology called HeliScopeCAGE. So CAGE stands for Capped Analysis of Gene Expression, and this is a single molecule sequencing method. There's no amplification step required, and basically what it does is it sequences the 5' cap of every single mRNA. So in this way, we can localize where a promoter, where transcription starts. And we can also use this in a quantitative fashion where the number of tags that map to a promoter start site or transcription start site reflects the degree of activation of that gene or the expression of that gene. So this is how we're looking at tissue-specific, tissue-wide gene expression. And just to give you an idea, we can look at expression across all of the different tissues that are in the Fantom5 CAGE data. And just to show you, here are two examples of a tissue-specific gene, which is ULK. ULK is involved in neuroblastoma, so germline mutations in ULK lead to a neuroblastoma. And you can see it's got this very peaked kind of expression that reflects the tissue specificity across the entire atlas. On the other hand, we have a more ubiquitously expressed gene, which is BRCA2, which we know also leads to cancer in the breast and the ovaries. And here you can see a much broader profile. The level of BRCA2 expression spans a wider range of gene expression values across the tissue atlas. So what we want to do is we want to identify the different classes of genes that are involved in tissue-specific or ubiquitous gene expression, as well as link those back to genes that we know cause cancer in a specific set of tumors versus more broadly in the human body. So what we've done is we've declared 4 different classes that speak to these different tissue-specific properties. We have class 1, which is the set of genes that have a mutation that causes tumors in a specific tissue and is expressed only in that tissue. We have class 2 genes, which reflect those genes that have a mutation causing tumors in a tissue-wide or ubiquitous manner, and they're also expressed across ubiquitous tissues. And finally, we have class 3, which represents our most interesting class, because this is a set of genes that cause cancer in a restricted set of tissues, but are expressed ubiquitously. So for instance, like BRCA1. And so based on our 76, I believe, germline mutation genes, we've classified which of those fall into our different tissue-specific groups. And let me just step back a little bit and explain how we came up with that classification. We've already come up with a tissue-specific penetrance definition. But what we also needed to do was come up with a way to define or classify which genes fell into the tissue-specific expression category versus the ubiquitous expression. And so this, as we saw with the contrasting profiles, is really a matter of declaring skewness. We saw that genes that have a very skewed profile, when we look at their expression in the entire Fantom5 tissue atlas, correspond to tissue-specific expression, whereas genes that have a much broader profile are more likely to be ubiquitous. So we solved this problem of identifying tissue-specific patterns by using mixture models. So we used the mClust-R package to fit mixtures, normal mixtures, based on the assumption of equal variance. And the goal is to try and capture the number of underlying tissue-specific or tissue-related states that are present for each gene. So just to give you an idea of what I mean, we fit for a gene expression profile, we fit a number of mixture models, depending on what the data says. And our rule is that if the critical regions from these separate mixtures overlap, then the gene is classified as ubiquitous. Otherwise, if we have more extreme mixtures such that they define specific sets of tissue-specific expression, then they're classified as tissue-specific. So this gives us a way to control for the error involved to a degree. And of course, what happens, you might ask, is if we have more than two mixtures. Well, in that case, we adopt or extend the rule such that for any two adjacent mixtures, these have to be non-overlapping in order to declare a gene to have a tissue-specific expression profile. So this is the rule that we've implemented to come up with our 4 classes. And as you can see here, these are the 4 classes, the genes that are involved in each of these 4 classes. We are most interested in class 3, primarily because this is the group of genes that represent the most potential for buffering effects to be occurring within regulatory networks. So one thing we've done is to really try to understand what these genes are doing and how they're contributing to this buffering role. If we step back and look at significant functional trends that are available or are detected in these gene sets, we see that ubiquitously expressed genes tend to be tumor suppressors, and that's based off looking at Go enrichment, CAGE enrichment analyses. We see terms that, like cell cycle, DNA repair, cell adhesion, come up, which are really characteristic of genes involved in tumor suppression. We've also looked at lists that have been annotated from the literature with regards to whether these genes are involved in tumor suppressor versus oncogenic functions. We also see for the tissue-specific expressed genes that they have Go and CAGE terms that speak to oncogenic function like proliferation, differentiation, migration, and survival. And the interesting thing is that class 3 genes are populated with tumor suppressor genes. So as I said, in terms of class 3 genes, our underlying hypothesis that we want to test is that tissue-specific networks are somehow buffering genes with germline mutations locally. So if you take, for instance, this is just a cartoon, but if you take, for instance, the BRCA1 network for a resistant tissue, which is not breast, not ovary, and you contrast that network with a network from a susceptible tissue like the breast or ovary, you would see local differences that in these networks that would confer some sort of buffering capability in the event of a germline mutation in BRCA1. So what we've done then is to construct protein-protein interaction networks, and we've done that using a resource called IRF Index. This is a database that combines all the information we have available with respect to protein-protein interactions. And there is a corresponding R package available called IRF-R. And we've also combined this information with tissue-specific resources. So there's a database called TIGER, which comes from Johns Hopkins, and it represents tissue-specific gene expression and regulatory information. And of course, we've also used the tissue atlas from Fantom5. So this is, these two networks are real PPI networks that we've reconstructed for the small intestine on the left and for the breast on the right. And the central protein, even though I know it's hard to see, is BRCA1. So it is difficult to make out topological differences just because there's so much going on in these networks. But there are differences that we're starting to see when we look at contrasting these networks directly. And so one of these differences is in looking at degree or connectivity of BRCA1 protein in these networks. And so what you see here is the degree distribution across all of the 40 different tissue-specific categories. And the yellow bars represent the degree of BRCA1 in the ovarian and breast tissue. And actually, the green bars represent the connectivity in corresponding cancer tissues. So we also had access to some different kinds of cancer lines and cancer samples in Fantom5. And so you can see, the other thing I should mention too is the 3 different lines correspond to the data that we have from the 2 different resources. So we have the degree distribution just from the TIGER networks, Fantom5, and by combining these 2 resources as well. So the immediate thing is that there is actually a lot of consistency. As we go from left to right, we see some differences in that number, but that number is not bouncing around. And certainly for ovarian and breast networks, there isn't a huge amount of difference compared to some of the others. But this graph does tell us that there are some extreme tissues on the end. I believe, for instance, like lung tissue, eye tissue, that there are differences in connectivity for the BRCA1 protein. So we can look more carefully at this network as well and look at what the other genes or gene products that BRCA1 is connected to, and look for functional roles for these genes as a set. And so when we do Go-and-KEGG enrichment, we see that some of the terms that bubble to the surface are related to DNA double-strand repair and mismatch repair. Now, this is not a hugely surprising result. We know that BRCA1 is involved in DNA damage response and obviously interacts with other proteins to form these complexes that are involved in DNA damage response. We are seeing this signature pop up both in this situation and other analyses as well, such as the class 3 set of genes themselves. And so it's leading us to think that DNA damage pathways are playing a role in normal tissues to set up some of this susceptibility to cancer that's occurring differently in different tissues. And this makes a lot of sense because we know that different tissues, even in a normal setting, are subject to different levels of DNA damage. So it makes a lot of sense that these tissues are using these DNA damage pathways differently, and such that in the presence of a germline mutation that affects all tissues, that you may see a different response, such that some tissues go on to develop cancer, whereas some others are able to remain resistant and be robust. So another thing we did to cut down the huge number of proteins that we're seeing in these PPI networks was to identify key components, or what we're calling gene expression hubs. And these are genes that when you compare their gene expression levels in breast tissue, normal breast tissue, compared to other resistant tissues, that there's a statistically significant difference at the 0.05 level, but also that we're seeing these genes are highly connected in the BRCA1 network. So we've used an arbitrary cutoff of 100 interactions, and when after this filtering, what we're left with is a smaller network, which I've shown here. And it might be hard to see, but there's some interesting genes that bubble to the surface. So for instance, HIF1 alpha, JAK1, these are all genes that have previously been involved in cancer. And so this, where it seems like they're involved here differentially in different tissues. So we've also done this with cancer tissue, and there's a mistake here in my slide. We haven't looked at normal breast tissue. What we've actually looked at is cancer breast tissue in normal resistant tissues. So those terms should be flipped. I apologize for that. But basically what we did was we took breast cancer tissue, breast cancer tissue, and we compared it to normal resistant tissues. And we looked again at genes that had a statistically significant difference in expression and also were highly connected in over 100 other interactions in the PPI network. And it's difficult to see the labels here in this network, but there are some interesting genes here, such as EGR1, TP73, and the androgen receptor as well. So we're in the process of exploring this further and talking to our cancer biology collaborators. So another thing we've done too is we've looked at some other networks, or begun to look at some other networks. Protein-protein interaction networks are useful, but they fail to capture some of the other more targeted regulatory relationships that we know exist. And one way of getting at that is to look at transcription factor networks. So we've used the network that's come out of MARA. MARA is a method for network reconstruction that's been developed by our Fantom5 collaborators. And they've used MARA on the Fantom5 tissue atlas to construct a global TF network. Within this global network, we've looked at connectivity that's associated with BRCA1 as well as other genes that have germline mutations. And what we've seen is there are, again, some differences that are apparent in these transcription factor networks between susceptible and resistant tissue-specific networks, but we're really only at the beginning of looking at what these differences are. Another thing we've done is to extend our classifiers to make predictions about what other genes in the human genome would be in terms of their tissue-specific classes. And so basically what we've done is we've used data in COSMIC, which is a genome-wide resource, as well as the information in the Fantom5 tissue atlas to predict sets of tissue-specific genes. And what we've done here is just shown the number of genes that fall into these classes, and we'll be making this available from our website soon. But the idea is to classify which genes might be involved in tissue-specific tumors as well as have tissue-specific expression. This provides a resource for trying to understand which genes might be more tissue ubiquitous versus tissue-specific. Finally, we've looked within these class predictions to look for any significant functional terms that are coming up. One thing that's been interesting within class 3 genes are those genes that are involved in tissue-specific tumors but are expressed ubiquitously, and they are potentially involved in buffering the response. One term that came up that was quite interesting was ubiquitination, and it could be that buffering mechanisms may be occurring through these kinds of mechanisms like ubiquitination. So that's something that we're interested in following up. I'd like to finish up my talk then and conclude with some of the results that we're seeing so far. So the goal was to understand why resistant tissues are able to buffer a germline mutation better than other susceptible tissues that go on to develop cancer. We looked for clues to answer this question in how tissue-specific networks control cancer genes in normal tissues, a range of normal tissues. We identified a set of class 3 genes. These are genes that have broad ubiquitous expression but are involved in a restricted subset of tissues. And we use these class 3 genes as candidates to look at local network changes that are occurring in normal tissues and to try and understand how different network configurations can contribute to cancer developing in some of these tissues versus others. For BRCA1, and it seems for other genes too, DNA damage pathways may be one of the distinguishing variables between resistant and susceptible tissues. And this makes a lot of sense given that different tissues in a normal setting are subject to or require DNA damage pathways, DNA damage response pathways, to differing degrees. So finally, we've looked at differentially expressed and highly connected nodes in these local tissue-specific networks in the protein-protein interaction networks, and they may be candidates for also contributing to driving this distinction. So I'm going to end there by acknowledging the people that we've been working with and the people who did this work. My postdoc, Dr. Danny Carbajo, has done largely all these analyses. We've also been working in collaboration with people at the Reconomics Science Center in Yokohama, specifically Dr. Alistair Forrest, Dr. Mikhail Dahoon, and also other people from the LSA Bioinformatics group. I'd like to end there by thanking again the organizers, Bioconference Live, for the invitation to be able to talk about our work. I'd like to thank you for your participation, your attention during this talk, and I'd just like to remind you that if you are interested in getting CME credits, the quiz is available, I believe, at this website, and I'd be happy and delighted to take any questions that you might have. So let me just have a look. Okay, so the first question we have is, have these type of analyses been made on mice models with germline mutations? One could manipulate them genetically, etc. That is a really great question. We haven't looked at mouse models. We've been looking at human data. I should just mention that the Fantom5 Tissue Atlas has both cell lines as well as primary tissues. Primary tissues have been collected from 3 different people with replicates, I believe. So there is a level of, I guess, natural variation there, but it certainly would be a lot cleaner and a lot more controlled to look at mouse models, particularly for these germline mutations, and as you mentioned, manipulate them genetically. I believe that for the 76 genes with germline mutations, there probably has been a lot of mouse work done on these genes. So it would be interesting to take that data and combine it and apply these kinds of analyses. But that's something we haven't had the opportunity to do so far. The second question that's been submitted is, how about comparing networks in breast tissue of very early onset women versus very late onset women with BRCA1? That is a fantastic question, and I think that really speaks to the next steps of why we're trying to do this work, is to be able to understand how it is that the robustness starts to break down, specifically because we're looking at normal tissue to begin with, and understand, how that robustness breaks down to lead to the development of cancer. I think the fact that we haven't been able to do that is largely because it's difficult to get access to that data, but that would certainly be very interesting to do that network comparison. And the final question is, were these comparisons done in silico analysis or in wet lab? This data was these comparisons were done on, I guess, in silico, in the sense that it's all been computational statistical analysis. We've used data that's come from wet lab approaches, but we haven't also been able to go back and validate them via experimental methods. So if there aren't any more questions, I'm going to end there, and thank you again for your participation and attention today.